Well, I want to thank Catherine and Mike for inviting me here, and really Mike for introducing me to Syngap community. It's so inspiring to be a part of this community and see all the incredible energy at so many different levels. And it's been really exciting seeing how this might fit in with my work and provide an avenue for some real therapeutic applications. So, I'm going to talk about really preliminary work. We've just started working on SYNGAP in my lab, and so take everything I say with a grain of salt, but hopefully you'll see why I consider this to be a really exciting direction that might have some really important therapeutic implications down the road. So I'm going to talk about a specific class of inhibitory neurons, parvalbumin interneurons, and gamma oscillations. I'm going to explain why I'm going to talk about those. I'm going to tell you about previous work in our lab, which has shown how important understanding these things are for thinking about therapeutic effects on cognition. I'm going to talk about a particular synapse we think is particularly important, and I'm going to explain how all this seems to fit together with SYNGAP in our preliminary data. So when I say parvalbumin interneurons, many of you will know this is a class of inhibitory neurons in the cortex, one of the largest classes, and they've been of interest in neurodevelopmental disorders for a few decades now, particularly based off of work in schizophrenia. So in schizophrenia, many researchers, especially at the University of Pittsburgh from Dave Lewis's lab, have looked at post-mortem brain tissue and seen that markers for this class of inhibitory neurons seem to be abnormal or reduced. So there's lower levels of the gene which actually encodes parvalbumin, the protein that marks these neurons, and then there's lower levels of GAD67, which is the enzyme which synthesizes GABA in these neurons. And so people have been interested in why these neurons are affected and what they might be doing in schizophrenia. And one thing that many studies, including some work that I did when I was a postdoc, have shown is that these neurons are really important for generating brain rhythms. And earlier in the talks on EEG, we heard a lot about brain rhythms, and brain rhythms are often named based off the frequency at which they occur, and parvalbumin interneurons are thought to be really important for generating these fast rhythms in the gamma frequency range, between like 30 and 80 cycles per second. Now, these gamma rhythms are often seen when individuals are doing cognitive tasks, especially the sorts of cognitive tasks that are impaired in conditions like schizophrenia and autism. So I won't describe the task because in the interest of time, but here is an example of power. So each of these like spots of red and yellow represents increases at a particular frequency, and this just shows that in this sort of cognitive task, which uses cognitive control, people have to stop doing an action they're used to and do a different action, you get a lot of gamma activity in EEG recordings over the frontal cortex, and this tends to be diminished in schizophrenia. And there have been many studies like this, both in schizophrenia as well as in other neurodevelopmental conditions like in autism. So putting together what I've told you so far, people have come up with this idea that maybe these neurons are essentially broken in neurodevelopmental disorders. As a result, they can't generate these brain rhythms, and maybe these brain rhythms are important for normal cognition. And this is a very attractive hypothesis, but it's been very controversial because even though it's easy to see these brain rhythms, it's been very hard and very controversial, the idea that these brain rhythms actually are important for brain function. Okay? And so it's been important to answer this question because we want to understand, like when we see deficits in gamma synchrony, for example, in schizophrenia, is this telling us that just like the brain tissue is abnormal and these oscillations are being generated in an abnormal way, but it's just sort of a marker that things are abnormal? Or is this actually a key process that we can go after and target to improve cognition in schizophrenia, but also in other neurodevelopmental disorders, including disorders related to SYNGAP, as I'll show later. Okay, so we've been studying this question by studying a cognitive task in mice, in which mice have to choose between different bowls to find a hidden food reward, like a tiny peanut butter chip. And each of these bowls has a different smell, so one might smell like garlic and the other like coriander, and each is filled with a different digging medium. So one might be filled with sand, and the other might be filled with wood chip. And mice will very quickly learn that one of these four cues, say the smell of garlic, indicates the location of the hidden food reward. And then we change the rule, obviously without telling the mouse. So we'll switch from, say, being an odor-based rule to being one based off of texture or vice versa. Okay? And this is the type of flexibility that we want to understand. What does it rely on, and how can we fix it in cases where individuals have become inflexible? Okay, and so I'm gonna make a few points. I'm gonna go into a lot of detailed neuroscience about this, but also just emphasize the take-home message. And the first key point I want to make is that these gamma rhythms are really important for flexible behavior. We've looked at this a couple of different ways. One thing we've done is to measure these gamma rhythms using what are called genetically encoded voltage sensors. These are proteins that fluoresce depending on whether a neuron is more or less active. And so we can measure fluorescence, measure rhythmic activity in specific groups of neurons, and then see how is that rhythmic activity synchronized. And basically what we found is that when mice are learning these rule shifts, that there's this big increase in gamma synchrony in the prefrontal cortex. And it specifically happens after error trials. And this is notable because when a mouse makes an error, that is the feedback that tells the mouse that it needs to update its behavior and start making decisions based off of a different cue. And right when a mouse gets that feedback, we see gamma synchrony start to go up. Okay, this is specifically in the gamma band, so we don't see this happening at higher or lower frequencies. Okay, we want to test the significance of this, and this will become important because we can then use these same methods to test the therapeutic potential of this approach. And for this, we've used something called optogenetics. Now, optogenetics is a widely used technique in neuroscience that will be familiar to all the scientists here and many others, but for those of you who are unfamiliar with it, I just want to tell a quick anecdote because it's such an amazing and revolutionary thing. Optogenetics was really developed and advanced in neuroscience in the lab where I was a postdoc in the mid-2000s, the lab of Carl Deisroth. And Carl, like me, is a psychiatrist, and we would actually see patients together. And one day, this was probably like 17, 18 years ago, patient came in, and he started off the appointment by saying, "Oh, Dr. Deisroth, I've been showing my friends YouTube videos about your research and telling them how you take genes from algae and put them in the brains of mice and then use lasers to control their thoughts." So this sounds totally wild and made up, but that's a perfect description of how optogenetics works. Optogenetics uses the tools of genetic engineering to pick specific types of neurons, make them sensitive to light, and then we can use lasers or other light sources to turn them on or off at specific times. So we've been able to use this in my research to study the role of these gamma rhythms by flashing light at 40 hertz to kind of weakly boost gamma rhythms in parvalbumin interneurons. And in particular, we can do so in different patterns. So we can deliver light to the left and the right prefrontal cortex that's actually misaligned and out of phase, so that these rhythms happen, but that the normal pattern of synchronization that seems to be important for learning a new rule is lost. And when we do this in previously normal mice that express channel rhodopsin, compared to controls, they take much longer to learn these rule shifts, and they become very perseverative. They become really stuck on the first rule. We've tried doing this with different patterns of light in order to disrupt other frequencies or even to boost activity and boost synchrony at 40 hertz. And it's really only when we disrupt synchrony around 40 hertz that we get this effect. So these brain rhythms are really important for mice to stop perseverating and to switch to the new rule. Okay, the second key point I want to make that's really motivated us exploring this in SYNGAP is that targeting gamma synchrony in this way can actually rescue cognition and rescue inflexible behavior. So we were studying mice that are heterozygous for DLX5 and 6. These are genes which are involved in the normal development of inhibitory neurons, and particularly important for the development of parvalbumin interneurons. And I'll just quickly mention these mice, they develop abnormalities in their parvalbumin interneurons. They have reduced output from parvalbumin interneurons. They're very perseverative in this task and have a hard time learning rule shifts. And we also see, when we measure electrically, deficits in task-evoked gamma oscillations. We can use voltage indicators and measure the difference between gamma synchrony on correct and incorrect trials. I already told you it's normally higher on incorrect trials. That's what we see when we look at wild-type mice, shown here in gray, and that normal gamma synchrony is deficient in these mutant mice. Okay, so they have broken parvalbumin interneurons and deficient gamma synchrony, and what is amazing is that we can fix them. So if we deliver different patterns of light in order to activate the parvalbumin interneurons in different ways, specifically when we deliver in-sync 40 hertz stimulation to parvalbumin interneurons in both the left and right prefrontal cortex, when we boost the normal synchronization, all of a sudden these mice start to learn rules just like normal mice. And what's quite remarkable is that this effect is persistent. So we'll do this stimulation one day, we'll test the mice using new cues the next day or a week later or a month later, and they'll continue to perform normally. It turns out that this remarkable kind of plasticity seems to depend on one particular synapse, and it's a synapse that people didn't even think existed a few years ago. It's a synapse which goes from parvalbumin neurons on one side of the brain to the other side of the brain. And if we inhibit just this synapse, we can make normal mice perseverative. That perseveration lasts. It takes them a long time to learn rule shifts the next day. Then if we stimulate just those synapses going from parvalbumin interneurons on one side of the brain to the other side of the brain, we can rescue their behavior, and that rescue is long-lasting. Okay? We also find that there are persistent changes in gamma synchrony, and actually if you look at how this task is being represented in the brain, that changes. So this brings me to SYNGAP. Is all of this relevant for SYNGAP? We think it should be, because there are deficits in parvalbumin interneurons in mice that lack one functional copy of SYNGAP. There's deficits in excitatory input that normally recruits these neurons and in the excitability of these neurons. And so I'll tell you now about our preliminary experiments, which again are very preliminary, but I think very exciting. This is all work that's been done by a great postdoc in my lab, Xiu Zhu. And basically what he found is that if you measure how long it takes SYNGAP heterozygous mice lacking one copy of SYNGAP, to learn these kinds of rule shifts from, say, garlic to sand, they too are very perseverative and take a long time to learn, as shown here in red. But if you now stimulate at 40 hertz in sync on both sides of the brain, the parvalbumin neurons, you can rescue this behavior, and it's rescued persistently. So you test them the next day, you test them a week later, and all of a sudden they're very flexible. They're able to switch normally. And what's even more remarkable is we don't have to stimulate all the parvalbumin neurons. We don't have to stimulate both sides of the brain. All we have to do is stimulate the connections going from parvalbumin neurons on one side of the brain to the other prefrontal cortex. And just that one synapse seems to be the key mediator of this procognitive effect. That made us wonder, well, why is it that this synapse is so important in these SYNGAP mutant mice? Is it because this synapse is broken normally? And that seems to be the case. Again, this is very early days and very preliminary data, and we have to expand this and do more controls. But when we've recorded from individual neurons and used optogenetics to stimulate these parvalbumin connections that go across the brain, what we find is that the strength of the synapses that they make goes down. It's lower in these SYNGAP mutant mice. All right, so that brings me to the end. Let me just summarize and talk about where we would see this going. These prefrontal parvalbumin neurons, this important class of inhibitory neurons, generates these rhythms in the gamma frequency band that can be seen on EEG, and we think this is very important for normal flexible behavior. We think that it's so important that if we restore gamma synchrony one time, we can actually rescue cognitive flexibility for a long period of time. This seems to depend on connections that go from parvalbumin interneurons on one side of the brain to the opposite side, to the other prefrontal cortex. And the reason that's the case, I don't have time to show this data, is because those synapses actually seem to undergo plasticity. Those synapses become stronger normally when you learn. They become weaker if you inhibit these synapses when mice are trying to learn. And so when we do this gamma frequency stimulation, what we found is that enables these callosal synapses to become stronger. Okay? And so our goal would obviously be to develop therapies, maybe identify drugs that might help these synapses become stronger. If you give that drug while a mouse is doing a cognitive task, maybe that would lead to kind of catalyzing potentiation of these synapses. And at least in DLX56 mice, we've been able to show that low doses of benzodiazepines, like clonazepam, which these drugs are often used at higher doses in SYNGAP-related disorders, they often have this effect of restoring gamma synchrony, restoring cognitive flexibility for a long period of time. So we think this is at least plausible. Maybe clonazepam won't work, but there might be other drugs which would. So again, I just want to thank all the people in my lab who have contributed to this work, and especially Xiu, who led this project. And of course, again, thank the foundation, not only for funding this work, but just really getting me turned on to this subject, which is a very exciting one, and hopefully will lead to even more exciting places.